Thank you, Caitlin.
Yep,
And,
sure, go ahead.
So thank you also for the invitation to speak, and thank you folks that are dialing in to be part of this. I appreciate it. I think the one thing that I still miss about this COVID situation is the personal interaction that you would normally get in a talk like this. Being able to look people in the eye and tell, if they're interested in what you're talking about or if they're getting bored. So it's, I think I'll do a little bit of a thought experiment here, and maybe in my imagination, I'm going to imagine that there's hundreds of people out there in the audience I'm looking at that are just riveted to their seats. And perhaps you could do the same and just make believe that I'm being interesting. Even if a few of you do want to fall asleep in the back row, that's okay too. I'll have my eye on you. So managing horseshoe crabs conservation at this time is important and also a little bit controversial. Let me stop my video here. We can just go to the presentation. To me, these animals are among the most beautiful and interesting animals on the planet. And it's kind of funny what you may think of as being beautiful. I can remember the first time that I actually saw a horseshoe crab. I was probably 6 years old. I grew up in the estuary in the Jersey Shore up around Shark River. And we spent our weekends on the boat. We had a little 15 horsepower wooden old town with a 30 horsepower Johnson on it. So we would be fishing or water skiing, and at lunchtime we would just pull into the estuary, way up into the river, and have some lunch. And I remember distinctly in ankle deep water, just being surprised by some large helmet-shaped crab coming toward my feet, and wouldn't have guessed that over half a century from then, I'd still be working with horseshoe crabs and have them play such a big role in my life, really. So let's see. Caitlin, are you there?
Yep.
Okay, I'm happy. Oh, here we go.
There you go. Yeah, your slide advanced.
Had a little trouble. So just an outline of some of the things I'd like to cover tonight. Obviously, on horseshoe crab, talk about a little bit of the distribution of their population, their unique immunology, the LEL, as Caitlin mentioned, and some of the conservation efforts that we're doing now. But then get to some of the concerns brought up during COVID. There was an interesting New York Times article about this, and caused a bit of a stir in the industry, and I could share our response to that. And then lastly, get to the issues of recombinant DNA technology and possible replacements or alternative tests, synthetic LEL, if you will. So first, the distribution. You can see here that horseshoe crabs really are in only a limited number of places on our planet. And in the blue on the left here, you can see this is the American horseshoe crab, Limulus, up and down the East Coast. And it's just limited to that. Nothing in South America, nothing in Africa, nothing in Europe. The Asian species shown in the purple and the orange, Tachypleus and Carcinoscorpius species, these are even more severely impacted by population loss than the American horseshoe crab. Very little regulation. They're collected for food as well as for bait. So we can certainly, I think, be aware of that and perhaps even participate in some activities to help them as well. Mentioning the LEL, that really stems from the unique immune system that the horseshoe crab has. And the basis of that is shown here. It's a cell that, when it attaches to surfaces like these few cells, they very much look like an amoeba, hence the name. So the important thing here is that you can see that they're loaded with granules in the cytoplasm. All these dark granules are chock full of enzymes that are part of the immune system. In the center, you can see the nucleus. But these are the cells that are collected in the manufacturing process to produce the LEL reagent. And really, what are they reacting to? For the horseshoe crab, they're certainly sensitive to bacteria in their environment. So if they were injured and bacteria got into the blood, they would react within minutes. And this is a schematic of the bacterial cell wall. At the bottom, you can see the greenish area is the cytoplasm, the cell membrane is in here, and then there's a cell wall outside that, and the external environment is up here. So part of the bacterial protective mechanism is this cell wall that's comprised of a compound called lipopolysaccharide, shown here. And this simply means it's a molecule comprised of both lipids and sugars or polysaccharides. And it so happens that in mammals, if our blood gets exposed to these compounds, it triggers our immune system, sometimes in a very severe way, to cause a fever. A little bit of reaction is actually a good thing. It stimulates our immune system to respond to that threat. And likewise, this is what happens in the horseshoe crab. But there's also a severe situation if we get too much endotoxin in our blood, that could actually cause toxic shock and death in certain circumstances. So the way that we can get that into our blood is if we, say, got an injection of a pharmaceutical, a drug that was prescribed to us, and it was contaminated with bacteria, then we run the risk of getting a potentially lethal fever from that reaction. So this is really the key utility of the LEL test. It keeps our pharmaceuticals safe for injection. And that's true of drugs as well as vaccines. So in the horseshoe crab, what happens when that lipopolysaccharide comes into contact with those cells we saw a minute ago? The enzymes inside those granules react to the LPS. And you can see here there are several enzymes. They're labeled, they're called factor C, factor B, pro-clotting enzyme. And when LPS binds to the first enzyme, it activates it. It becomes active on the second enzyme to activate it. And likewise, that one in turn reacts a third enzyme. And each step along the way amplifies that situation about tenfold. So a tiny amount of lipopolysaccharide can have an enormous number of activated enzymes down here. And the final step is those enzymes work on a protein called coagulagen that becomes a clot. So again, for the horseshoe crab, this is their immune system. Very simple, very fast. Within minutes, this happens. And that clot would serve to seal the wound, trap the bacteria, and allow them to survive and live. So we even think that this may be part of the reason the horseshoe crabs have survived so long, over 450 million years, throughout the evolution of the Earth. Now, I'm going to also talk later on in the presentation about synthetic alternatives to this. And while I have this slide up, I wanted to mention these enzymes again. And one, this factor C, was the first gene that was isolated and cloned from a horseshoe crab. And with the idea that factor C alone could serve as an assay for the LPS, for the endotoxin, simply by being activated and then using a colored reagent that reacts with this enzyme as a test. So that's one. And then also, we want to consider making and cloning the genes for the entire cascade. So having factor B, factor C, as well as proclotting enzyme together cloned so that you could have a total synthetic LEL cascade. And I'll talk a little bit about that later. So now just to some of the things that we're currently doing for horseshoe crab conservation. Just a horrendous picture from the 1930s and 40s where the species, this is well before the discovery of the biomedical use of horseshoe crabs. And this shows just the quantity that were consumed for fertilizer and for bait. Just something we wouldn't imagine of doing these days. Now, we do have the Atlantic States Marine Fisheries that takes really close tabs on the horseshoe crab population along the East Coast. And in this graph, this shows multiple years going back to 1998, showing the number of crabs that were landed. Okay, so in this case, the orangish color is signifying those landings for bait. So between 2.5 and 3 million crabs, you can see back in '98 were collected. That is here showing year by year going further down as more and more states outlaw the process of collecting horseshoe crabs for bait. All of those crabs collected, of course, die. So it does have a direct impact on the population and the survival of the species. So beginning around 2002 to 2004, they started also quantifying how many crabs were landed for LEL production, the biomedical application. And in this case, these again are just landings, they are not deaths. So to estimate the number that could be dying due to collecting the crabs for the LEL industry, they really take a guess, an estimate of 15% loss. And that's shown in the darker color here at the bottom. Sorry, having a bit of trouble with my cursor. Okay, so these are the landings, and then you can see the deaths here. And it's been relatively steady over the past decade or so. And again, with the collection and the deaths due to bait steadily declining. There's another way to look at that, which is to break up the East Coast into divisions. So Northeast, New York, Delaware Bay, Southeast, and then coastal wide. And then to judge when they do their trawls to count the horseshoe crabs, do they see those numbers declining or staying the same or increasing? And obviously, if it's declining, it's a poor situation, labeled in red. And traditionally, the Northeast has been really the worst, northern regions. However, that has been improving, from 2009 to 2019, over 10 years. There's been a lot more awareness of the potential loss of horseshoe crabs, a lot more care taken. Again, less bait industry being allowed to collect in many states. So that's having a positive effect. So now some of the worst areas are labeled as neutral. The one that is still continuing to be poor is the area around New York. So Long Island Sound in particular, really due to, I think, more about habitat loss than anything else, but certainly something to be concerned about. Delaware Bay also is labeled neutral, but that's an improvement over the earlier days of last century, especially when the population really declined quite a bit. One of the best areas actually is the Southeast. This is where Charles River collects horseshoe crabs for the production of LEL for us. And the situation there, the environment obviously is a little bit different than a more heavily populated Northeast, which helps, but also the estuaries in the Southeast around Charleston and Georgia, Virginia are just enormous compared to maybe more beach atmosphere in the Northeast. So there was a recent paper out talking about the very large contribution of these estuaries for immature crabs as a haven for the population that could explain also the goods population maintenance here. Also in South Carolina, when our company was formed, we actually lobbied the state to put laws in place to protect the horseshoe crab and to limit the collection only for LEL production. And they take extreme rigorous care about keeping track of us. We're pretty highly regulated there, and that's not a bad thing.
Hey, Norm?
Yeah.
There's a question in the chat box. In New Jersey, how many exactly are collected, landed in a figure? So is there a figure for how many are collected and landed in New Jersey, horseshoe crabs?
Yeah, I don't have a specific one for New Jersey, but I think in New Jersey there really is a very limited collection for LEL production. Charles River actually buys a few from New Jersey, but by far, a much higher amount, by a factor of perhaps 4 or 5, is the number of crabs we get from South Carolina.
So they are collected in New Jersey and bled, right, for LEL, but it's very limited, correct?
Yes, that's right.
Okay.
Also by state law, I believe.
Right. Okay. And if I didn't answer, or maybe I didn't, if I misconstrued that question a little bit, just let me know in the chat, but I don't think there are any other questions right now. So I'll let you go ahead.
Okay. So the other way you can conserve is to use less. And this actually shows technology that we developed. And there may be some of you in the audience that saw a talk I gave a couple of years ago in this forum, where we actually flew this miniaturized version of the LEL test to the space station for measuring bacterial contamination on board spacecraft. And in this case, this is a cartridge along the bottom, and it's about 1 inch by 4 inches. So rather about the size of a microscope slide. And on it, there are 4 channels that we actually put a very microscopic amount of the LEL reagent in there. And then this cartridge slips into a reader. You see the tip of the cartridge down here. So in the reader, there's actually a pump that will pull the sample in. The sample would be put in right here. And it only takes about a drop of sample, 25 microliters. So when all 4 channels are loaded, the pump will pull the material in. It will rehydrate these reagents, the LEL reagent. There's a chromogenic substrate that changes color when the reaction occurs, and then it gets read in a little optical cell here. So in the instrument, there's a light source and a sensor, and it's like a little computer that calculates the amount. But it has an enormous impact, not only on the ease of use, but it uses 95% less of the reagent. So this is just to show schematically the traditional LEL. If we were using, 5 liters of LEL, it's over a gallon, that the current test with the miniaturized chip would only use about a cup and to do the same amount of tests. So that has an enormous impact on conservation as well. The other thing that we're doing is actually a partnership with Rutgers. Several years ago, we established a partnership to collaborate with Rutgers collecting fertilized eggs from the environment, bringing them into the lab, and then growing them, feeding them over the course of several months to allow them to become more mature, and then get released back into the environment. And the idea is, as a more mature young juvenile, that they would survive much better in the environment than the fertilized eggs, which are eaten by many species, by birds, by fish, and just natural losses. So this is a way that we can try to enrich the populations, and it's something that we're actually trying to work with other collaborators up the coast. So in addition to Rutgers being the first, we now have replicated that technology at Mystic Aquarium and Marine Biological Lab in Woods Hole. South Carolina Aquarium is also interested in participating. So we hope this grows and becomes something that we can actually establish as a concern to help bolster the populations up and down the East Coast. And just recently, we were actually contacted by some folks in China to see if we could export this technology and help save the Asian species by this method as well. So now to the COVID situation, and I cringe when I see this picture because I hate needles. I'm sure some of you do too. But the fact is, there just is an enormous amount of our pharmaceuticals that are tested and kept safe by this LEL test. And the number here of 35 billion doses of vaccines, that is not just the COVID vaccines. These are routine vaccines that are produced every year. So just something to be aware of. And sometimes we are not so aware that these things happen in the background, but and the contribution of the horseshoe crab to that is really enormous. And just a word, I guess, about that too, is I was just thinking how ironic it is for the horseshoe crab, who has that primitive immune system. Their primitive immune system is the LEL. They don't have antibodies, so they could never benefit from all these tests that they maintain as safe for us to use. But it is something to think about. There was some controversy starting probably last summer. And let's see if I have it here. Before I get to that, I forgot, I wanted to mention a little bit of history of testing for endotoxin before horseshoe crabs became popular. So in '83 is when horseshoe crabs were officially recognized by the FDA for this purpose. But back in the earlier days of the FDA, in 1940s, rabbits were used. And the way that test works is several rabbits would be injected with a drug, a sample, and then their fever would be monitored, their temperature would be monitored. And if they did rise in temperature, have a fever, that was a bad thing and a positive test. So this test was not very quantitative and frankly, not as reliable as it could be. So the FDA was actually in the 1970s an active partner with scientists around the country to develop the LEL industry, to develop specifications for the test, and then again, the approval in '83. So by '87, widely accepted and really the industry standard for testing endotoxins. I mentioned that cartridge that we introduced, and that was first accepted by the FDA in 2006. In 2003 was the first product using a recombinant factor C. That's that limited recombinant with one enzyme cloned. And this was not approved by the FDA and still has not been, but it is an experimental use and has been used by many researchers around the world. So to the controversy, this came out in the New York Times in, I believe it was June last summer. And as the title says, tests for coronavirus vaccine need this ingredient, horseshoe crabs. And it was an article that was heavily weighted toward some conservation groups that were warning that the number of LEL tests needed for billions of COVID-19 vaccines would outpace the natural horseshoe crab population and really cause a damage to the species, to the population, as well as put at risk all those tests that are needed for the safety of the vaccine. So their positioning was that we should move immediately to this recombinant factor C as a synthetic alternative. And there was also a bit of concern that both European and US regulators wanted more time to prove that the factor C synthetic was good enough, was as good as the natural product. So it was being delayed, and basically it was an article complaining about that delay and complaining, why don't we go to the recombinant? And this is also going to hurt the crabs. Well, that resulted in quite a stir within the LEL industry. And I don't expect you to read this letter. I showed it only because it's signed at the bottom by the leads of 3 different LEL manufacturers, the major manufacturers that produce LEL in the world. Our company was one of them, Associates of Cape Cod and Luanza. And this would be sort of the equivalent of Ford and Chrysler and GM putting out a joint press release. It really is something you don't see very often. But I think it was in response to that editorial and the fact that it was very misleading and maybe containing information that really wasn't accurate. So what they did in the text of this, I'll blow it up so we can see, they actually did a calculation of if, for example, we wanted 5 billion doses of COVID and the batch size for vaccines is about 100,000 doses, which is about, it was not uncommon for most vaccines. Then just doing the math, that means there would be 50,000 batches of 100,000 doses to meet that need. Well, the way the industry does the testing is it takes 3 vials from each of those batches, one from the beginning, one from the middle, one from the end, as the regulations stipulate. So that means of the 50,000 batches, that's now 150,000 tests are needed. So one more, bear with me to go through this math. So if you have 150,000 tests you need, you do the test in quadruplicate. So that means times 4. So we have now the number is 600,000 tests that have to be done. And when we make LEL, typically it's done in a 50 test vial. So dividing 600,000 by 50 tests, you come up with about 12,000 vials of LEL that's needed to test all of the COVID-19 vaccines that would be produced. So what really irked, I think, the manufacturers, including us, is that amount of vials, 12,000, is less than a day's combined production for all 3 of these LEL manufacturers in the United States. In other words, it's basically kind of a drop in the bucket. So that's one concern that, you know, certainly the industry was concerned that we would be kind of mislabeled as using billions of LEL tests and costing the lives of many horseshoe crabs unnecessarily. It's just not the case. So now let's go to the issue of, could we come up with a recombinant DNA answer to the future supply of endotoxin tests? And again, I just want to remind you of those two approaches I mentioned. That first one is to clone just the first enzyme in the cascade, factor C. And as I mentioned, that has been done. There are commercial products on the market that do this. Approach two would be to clone the entire cascade and try to get the advantage of that amplification that you get from multiple enzymes acting together. So we, as a company, decided to do a study. And that study was just recently published in the European Journal of Pharmaceutical Sciences. The title you see here is The Evaluation of the Limulus amoebocyte lysate and Recombinant Endotoxin Alternative Assays for the Assessment of Endotoxin Detection Specificity. In short, what this is really a comparison between natural LEL and some of the recombinant products to assess just how good do these products compare. And our method was to basically enlist all the pharmaceutical manufacturers. And I'll go through what we found here in kind of an abbreviated way. What we reasoned was, and what we asked all these manufacturers around the world, went over 30 countries, we got 128 samples of water that is used as the feed water for their purified water systems. And I hope I didn't mix you up there, but when you do pharmaceutical manufacturing, you have to use very, very clean materials that are certified free of endotoxin. The water going into all of the solutions has to be free of endotoxin. So any endotoxin contamination that's detected at the end could be coming from any of these sources. So the 128 samples we received are shown on this graph. You know, the higher the number, the more endotoxin. So low down here, sorry, high up there. And we compared not just our material, Charles River. Charles River material is on this axis, and our competitors, Lonza is another company, is on this axis. And this was a beautiful thing because two separate companies, two separate regulatory mechanisms, all regulated by the one FDA. And what you see is almost an exact correlation of samples, the values of all these 128 samples. You see the statistics up above there. The correlation coefficient is almost perfect. So we took those same samples and we tested 3 factors, recombinant factor C products, and they're shown here. These first 3, one's named Pyrogene, Endozyme 2, and another Endozyme 2 Go. Again, all 128 samples, and the alarming thing here is that the number of samples where the value was less than 1/2 of that calculated by, you know, the LEL test is shown in this column. So of 128 samples, 100 of them were 50% less for this particular product. The next one was even worse, 122 were less. The third one was a little better. This is a newer product, so probably is going through some improvements, but still 83 samples were less than half of that predicted by the LEL reaction. And just a word of why this might be important. These endotoxins are coming from naturally contaminated water, meaning that these are the endotoxins that, if there was ever a problem with the water, could seep into the product. Uh, when an endotoxin test is done in the lab, say for research purposes, many times the standard endotoxin you would use would be highly purified. So many of these products compare purified endotoxins of the recombinant factor C test to the natural LEL test, and they come out pretty good. But when these products see the natural endotoxin, there seems to be an underprediction of that value. This is another way of showing the same information. These are the two LEL, natural LEL, one's Charles River, one's Lonza. And then these three are the Factor C test. And again, you see the error bars and the amount of decrease in the detectable endotoxin. What that means for a product is that if you're using something that underpredicts, then you could have a contamination that falls outside the sensitivity of that product for that natural endotoxin, and therefore it would be seen as a negative, even though there would be endotoxin in the sample, and that would put patients receiving that at risk. Let me go back to this graph one more time, and there was another line here labeled RLEL. This means recombinant LEL. And this is actually an LEL product that we make that is totally synthetic as well. So we cloned all three of those genes, made a mixture as an experimental first attempt at a recombinant. And we were not happy with it either. Of the 128 samples, it's still missed 50 that were below 50% of the natural. It was significantly better than certainly some of the recombinant factor C only, but we still decided that this is not ready for prime time yet, and we are continuing to work on this. This data is about 6 months old, and in fact, we are improving this significantly, but it has not been proven yet. And again, on the graph below, here's where the recombinant LEL falls. Again, a little bit better than the recombinant factor C, not quite up to the standard of natural endotoxin.
Hey, Norm.
So, yeah.
We do have some questions in the chat. One of them being if there's a link to this report, if you happen to have it, if it can be placed in the chat box, or it could be provided through email. If you don't have it on you right now, maybe we can do through email of the participants as well. But do you have it on you to pop in the chat right now, or maybe at the end of the talk?
Perhaps at the end.
Sure.
I don't have it handy exactly, but if not, I can send it to you by email if we somehow miss the chat, and we can get it out to the folks.
Yeah, that sounds good. And then I wasn't sure how many more slides you had left. There were some questions. Did you want to answer them now or wait till the end? I
have just one more slide. I was going to do a conclusion.
Okay, I'll let you finish up and then we'll get to the rest.
Yeah, so this is our thinking at the present time. This is not an official conclusion by any regulatory body or anything like that. This is our opinion. Number one, for now, continue relying on the natural LEL for patient safety. Number two, keep improving the recombinant technology to equal the natural LEL safety performance before making a switch to synthetic alternatives. And I have full confidence that we will get here relatively soon. And number three, allow the FDA to regulate the new recombinant DNA alternatives to LEL when they're proven safe. And I think the point here is that you noticed the two competitors for the natural LEL, both Charles River and Lonza, how close they were to each other is astounding. And I think that's a tribute to the fact that the FDA oversees this. They extremely tight reins on our production methods. We're inspected by them all the time. So that's a good thing. It's a pain in the butt sometimes, but it's also good in terms of patient safety information. So yeah, I can stop there and we can take some questions or comments.
All right. Well, thank you very much. Very interesting. We do have a bunch of questions in the chat box. I'm going to put my camera back on. I'm going to go back up in the chat and start with one about moratorium on the number of landings in South Carolina. Is there a moratorium? We know we have one in New Jersey, but someone was wondering if they could know the exact number of horseshoe crabs that are landing in New Jersey each year.
You talking New Jersey or South Carolina?
Oh, sorry. The first part was if they were wondering if there's a moratorium in South Carolina. The second part of the question is how many are landing in New Jersey each year?
No, there's not a moratorium now. There was an attempt, there was a lawsuit that was filed. Some of our collection was actually done on federal land. So a conservation group brought a suit to federal court. And initially they ruled in favor of putting moratorium in,
Right.
but then the state of South Carolina actually stepped in on our side, basically, and put a stay to that. So, you know, that doesn't affect the entire state, just a small portion of where we get our crabs, but there will be no moratorium this year, apparently.
Okay. And do you know how many are landed in New Jersey each year? If not, I can provide the link to the American, the Atlantic States Marine Fisheries Commission about horseshoe crabs. I know there's some stock assessment reports that could help answer this question.
I think on the, I can't answer the question, but I think on that site, they do break out those numbers by state or certainly by region that we could figure out approximately how many are in New Jersey.
Yeah. I think if you go to that link and then you scroll down, there's a bunch of stock assessment reports, which can probably help you answer that question to exactly how many are landed in New Jersey each year. So hope that helps. The next question is, why is there a 15% loss of horseshoe crabs after the landings and LEL harvest? And what is being done to reduce this high level?
Yeah, well, that's actually not a hard number. That's an estimate. And we actually know what our losses are in South Carolina, and they're much less than that. They're in the range of, I believe, 6 to 7%. But the losses are due mostly to the handling. So we take pretty good care on both ends. That is, we collect them by hand, not through trawling. So they're placed into a boat. Eventually they go into a barrel that's kept moist and covered in the shade. And then it goes back in the water, within a day. So those, if there are losses, that's where they're lost. There's also some science that's being done saying that the bleeding process itself could weaken the animals so that they perhaps have diminished breeding capability. I understand that science, and I don't fault it. They're looking at certain biomarkers in the immune system. But I go back to the Atlantic Marine Fisheries report again, and Department of Natural Resources in South Carolina, that our population is actually stable and growing. So we've been doing this for 30, 40 years and keeping pretty good control of how we handle the animals, and it's having no ill effects. So I think those are minimal. It will never be zero. And I think that's the cost of the benefit of this technology, of any technology, is going to have a downside. And even though we try to minimize it, it will happen.
Mhm. Okay, great, thank you. And then what are the challenges, opportunities, and time required for synthetic or recombinant LEL technology to be approved and widely employed by biomedical and pharmaceutical industries?
Yeah, there's two things there. Number one, you saw a little bit of it when we did our first approach with the totally synthetic. It wasn't quite good enough. So that takes a little tweaking, meaning how much of each enzyme do you add? What's the formulation, the pH, the buffer? We've had decades to optimize the natural product. So it's not going to take decades, but there will be some experimental formulation that takes to really get it improved. The other thing is making recombinant enzymes that are free of endotoxin is not easy. So that has taken a while to figure out. And the third thing is the regulatory aspect of it. It really won't be fully adopted until the regulatory agencies put their stamp of approval on it. And that means it has to be shown to be as good as the natural, that there's no risk to the patients. And again, I hope that happens soon.
Right. All right. And are there age, size, and sex requirements on the horseshoe crabs that can be landed and bloodlet for biomedical research?
Actually, I believe no. The ones in South Carolina, at least, the adult animals are all together, and they can both male and female can be taken. As you know, females are significantly bigger than males, so they do have a little bit more blood, but they perform equally well.
Okay. And then kind of related to the question we did prior, do you anticipate the FDA will approve the recombinant factor C? It sort of relates to the one that you answered 2 questions ago.
Yeah, well, I didn't underestimate what I said. This is very controversial, and with that paper, and I'll send that link to the paper, we raised doubts that the recombinant factor C formulations that are out there could pose a risk to patients, and that's not something that we could stand, both ethically and legally. That would just not be in the nature of us to do. So that, on the other hand, we're very much in favor. You can imagine making the product recombinantly is going to be, you'll have a lot more control over the process. The natural blood that you collect every year is from batch to batch is different, which creates a nightmare to formulate a consistent product. So we can do better with recombinant. And I think it really relies on, depends on the fact that the regulatory agencies will also accept that and to move in that direction.
All right, and then back to our, with the bloodletting and landings of horseshoe crabs, how many horseshoe crabs need to be landed to provide the 12,000 vials of LEL, and how many of these horseshoe crabs are lost due to this bloodletting? I'm not sure if that the 15% is included, maybe in that last part of the question, but
yeah, I'm not sure I could answer that question. Not because I wouldn't want to, but doing the calculation of how much LEL blood would basically be collected to produce 12,000 vials, or even 1 vial. I'm not sure what that number would be. It wouldn't be many in the scheme of things. I think industry-wide, I think the numbers that are collected are maybe 300 to 400,000 per year. So yeah, it would just be a handful, I would think, to make those 12,000.
Okay. How much blood can you it comes from? What's is it like, quarter of a liter? How much blood actually comes out?
Yeah, it depends on the size, and again, the females are a lot bigger, but it's in the neighborhood of 200, 300 milliliters.
Okay, wow.
So about it's significant. It's maybe about a cup, a
Hmm.
little bit more.
That's a
per
safe
animal.
amount even, yeah.
Well, I mean, again, the blood is not actively taken out. It's taken, their circulatory system is under low pressure. So a needle is inserted, and then that's how much drains. And then it stops draining, and 2/3 or more of their blood stays with them. And I think that's probably why they survive so well
Mhm.
through that process.
And what happens to the landed horseshoe crabs after you bloodlet them? Do you observe them, nurture them, and then release them back to where they were landed? Where is the 15% loss occurring in this process? But I think you may have answered that question earlier already.
Yeah, well, I think so the answer is that they go back in the water. That's the prime mission. As soon as their blood is collected, they go back in the barrels, kept moist, covered with burlap in shade, and then go back into the water, where they were, near where they were collected.
Gotcha.
They do, of course, try to minimize recollecting the same ones. So, there's the collectors will move from spot to spot, and through the years, if they see it has been bled, they won't take it again.
Gotcha. Right.
There's usually a little bit of a scar where the needle is where it was inserted.
Okay. So they can tell when a crab has been recently. Okay. Interesting. Right. And then I think this is our final question for the evening, which is actually right on time. So it's kind of neat. Which assay is on the market first, yours or Lonza?
Well, this goes way back to the beginning of the industry. So I believe the first might have been Associates of Cape Cod. I think Charles River was called a different name then, called Endosafe. That was number 2. And then there was Lonza was, again, a different company name, but I think they were number 3. And that was back in the 1970s, starting around 74 to 79.
Okay. And then there was another comment about potentially maybe not seeing the exact landing numbers for, is it biomedical research only? I wonder, are horseshoe crabs managed by the Mid-Atlantic? Could we get information about landings through the Mid-Atlantic States Marine Fisheries Commission? Or maybe I'm saying that organization wrong. I'm just trying to see where we can find reports if people are interested in finding more information about landing reports.
Yeah, I'm not sure, but when I look up the link to that paper, I'll put out the question to some other folks in our company who might know.
Okay. All right. I don't think that's, I mean, I do think that that's the last question. There doesn't look like to be any other ones for this evening. I want to give you a virtual round of applause, Norm. That was great. So we can do that. If you feel like adding a hand clapping to reactions, you can do that too.
All right.
Everybody
I appreciate
wants to.
that. Thank you.
I know we have to, do our round of applauses differently now, but I want to extend my appreciation again for you taking the time out of your evening to do this very relevant talk. And I have everybody's contact information. So when Norm finds that article or asks, for more information about landing numbers, we can provide that to you in a separate email. So
actually, Caitlin,
yes.
One thing I could do is I have a PDF of that on my computer, so I could just email you the PDF and you could forward that. It might be easier.
Okay, yeah, whatever works. That's great.
Okay, will do.
All right, everybody. Well, thank you so much once again for attending our lunch and learn, or sorry, our eco evening. I've got lunch and learn on the brain. That's tomorrow. And enjoy the rest of your night tonight.
Yeah, thanks, Kate.
All right. Yep, thank you, Norm. Thank you, everybody.